No math. That's the answer.
Okay, all right.
Yeah. Okay.
We're still waiting for speaker, is that right?
Yeah.
Yeah, I haven't seen the something on.
I exchanged emails with him this morning, so he'll be on here in a few minutes.
Okay.
I will, Tom, maybe you should make John a co-host.
Yeah.
And Zhigang as well.
Yeah.
So they can help promote panelists.
So John and Zhigang, now you are co-host, so if you see
Mhm.
any familiar faces from the attendees, please feel free to promote them to the panel. And attendees.
Why it's stopped? Here it started.
So Jimmy, I think we're all set to go with Takao, right? He's next.
Correct. And at the end of this webinar, we're going to promote that webinar.
Okay, our speaker.
Let's call it part 2 of a mini series.
Okay, great.
And for the announcement today, I also included a few recent papers that we want to promote and include information about EML webinar overview.
So do you want to start by going through these slides, Jimmy, or do you want to do this after the discussion interview?
Perhaps we can do it about 5 minutes before the start of this webinar.
Okay.
Since our speaker is here, should I stop the slideshow for him to try his slides?
Yes.
Yeah, I think so.
Okay,
Hey,
let
Dae-Hyeong.
me stop sharing the.
Morning.
Good evening. Dae-Hyeong.
Hi,
Hey, Dae-Hyeong.
good to see you.
Hello.
Yeah, let me try to share my screen then.
Sure.
Yeah.
Go ahead.
Do you have videos? You probably want
yes.
to run. Okay. Those are the most challenging ones. We can see
Jimmy,
your slides.
what is the official time of start? This you said this starting time is special. What is
Right.
the starting time?
9:30.
Oh, 9:30 start.
Yeah, we
Right.
started half an hour earlier today.
Okay. 9:30 is already half hour. I'm confused now. So now we are very early now, right? 9:30 start. Okay.
Right. And actually, next week for Takao seminar, we're going to start at 9 o'clock Eastern.
Okay.
You mean the webinar itself?
Webinar itself, right?
Oh,
so
okay.
still
Okay.
half an hour earlier, he requested it because in Japan it would be 10 p.m.
Yeah, that's right.
Yeah, it seems that everything is working right properly.
Great, great.
I guess.
Wonderful.
Okay.
Yeah.
So maybe we can start the informal interview now. John, you want to start?
Yeah, sure, I'll do that. So welcome everyone who's on the call to our EML webinar today. We have speaker from Seoul National University, Professor Dae-Hyeong Kim. He's a close friend and colleague that I've known for many years, and we'd like to welcome him as our speaker today. And so I think we have about 30 minutes, during which time we'll just have an informal discussion, and we'll interview Dae-Hyeong, ask him some questions about his career trajectory, his research interests, maybe some personal questions as well, to the extent that he'd be willing to answer those. So we'll just kind of open it up. And I also want to welcome Ting Lee and Jimmy Shaw and Jigang Suo, co-editors, lead editors at EML, who are on the call as well. So welcome, Dae-Hyeong. It's great to have you as our speaker today.
Good to see you, everyone.
Yeah.
Thank you very much for a kind invitation. It's my pleasure.
Yeah, I very much looked forward to hearing the latest, research from your group. You always give a great talk and lots of really novel ideas and interesting material science topics, but mechanical engineering also plays a prominent role, I know, in your research. And so I think these topics will be very well aligned to the readership at EML and more broadly as well, cutting across many different traditional disciplines in engineering and science. And so that's great. So full disclosure, I sent Dae-Hyeong some questions that I plan to ask him now. So he should be somewhat prepared, but I think this is, it's going to be informal. So, Jimmy and Jigang, Ting, feel free to jump in and ask questions if you'd like. But maybe we just get started. Dae-Hyeong, you could tell us a little bit about your overall career trajectory, maybe give us some stories about how you first became interested in science and engineering and what led you to, this particular choice of career as an academic and a leader of a really vibrant and robust research group. So maybe you can just say a few words about your background.
Yeah, I thought about that, and I feel that my parents recommended me to choose science high school. That was the first that I was interested in science and engineering. Actually, during the science high school period, I could have many chances to have experiences in experimental physics and chemistry. So naturally, I became interested in the engineering school and the natural science. And I chose, more specifically, chemical and biological engineering department, which I belong right now. That was the start. But more important event that I chose the academic position is due to the valuable experience and education that I could have in the University of Illinois during my PhD and postdoc period under Professor John Rogers' advice. I learned the importance and the delightfulness to work with collaborators and make new inventions, which were mostly thanks to kind helps and advices from my advisor.
Well, those are some kind words. I'm sure you're giving me way too much credit. I mean, I kind of hit the jackpot when Dae-Hyeong decided to join my group, and he went off and operated in a very independent manner almost from day one. And then I just had the pleasure of watching all these great research results coming out of his efforts in the lab. So it was a wonderful experience having you in the group. I mean, one of the talents that you have really is the ability to do research in an interdisciplinary mode, and you seem to be able to collaborate very effectively with people in disparate fields of study, neuroscience, medicine. I think we're going to hear a lot about that in your talk. For people on the call, what are the keys to being successful in that kind of collaborative mode? Do you have any sort of special approaches that you use to be effective in that way? Not all people are skilled in that sense. Are there things that you can learn to be more effective in that manner?
I personally try to communicate with other researchers, especially in clinical field, as often as possible through conferences or even private meetings. Especially for the biomedical researchers, I think that figuring out the unmet clinical needs is very important. As far as we can understand the unmet clinical needs, we can accurately and properly design materials and devices, of course, in consideration of the mechanics. Mechanics is always very important in material preparation and also device designs. So, and then we frequently and periodically discuss with our collaborators, both in the clinical field and also engineering mechanics, to improve the device performance and also optimize the device function for specific clinical application.
Yeah, that makes sense. I guess kind of a related question is, how you go about choosing research topics to pursue. So you're operating at this very vibrant interface between engineering and medicine. And so of the current projects you have going on in your group in that space, how many of them were sort of brought to you by clinicians who are grappling with unsolved problems or challenges or unmet needs in patient care? And how many of those projects really bubbled up more as a result of open-ended kind of exploratory academic research where you subsequently found, some kind of medical need that could be addressed with that technology? Is there some kind of blend there, or how do you approach, launching new projects in your group?
Mostly such projects were initiated by ideas from clinical fields. So I belong to Institute of Basic Science, and under that umbrella, we have a close collaboration with clinicians in cardiac field, as well as tumor, cancer, and also brain research field. So there are many clinical collaborators, and they always bring new ideas, and also they frequently visit conferences in their own field, and they bring new ideas and new concepts to us, and that initiate the research products in biomedical areas. But at the same time, my students are eager to study new biomedical device and material ideas, including hydrogels and conductive nanocomposites and stretchable, flexible biomedical devices. And also, we are very much inspired from the research in John Rogers' group, which is always inspiring and pioneering new fields. So yeah.
You're coming along.
I personally think my collaborators and students.
It's a little embarrassing. You're coming up with a lot of really great answers here, Dae-Hyeong. Good job with this. Just kidding, obviously. So I guess a lot of people in engineering science would like to have more collaborations with the medical community, and I think some kind of struggle with making that happen. And so, are you spending time on the medical school campus, or are you hosting clinicians in your labs, or what are the mechanisms that are driving that dialogue, which, as you mentioned, is so important in sort of identifying meaningful research projects in your group? Or do you ask your students to take courses through the medical school, or what are the different mechanisms by which that happens, those interactions?
So I had personal experiences in medical school when I was in Illinois. I visited the UPenn for a month, and at that time it was very beneficial. I could go through the clinical procedures as well as all the animal experiments. That was very helpful for me personally. So I did the same to my students. Luckily, Seoul National University Hospital is quite close from our campus, so just on an hour by public transportation. So my students frequently visit Seoul National University Hospital to perform animal experiments and also watch clinical procedures. So and during that period, naturally they have very deep discussion with the clinicians, and even during lunch or dinner, they can make new ideas and spark with the new concepts. So, yeah, I think that is a very good education for the students.
Yeah, that's a great description, and hopefully, that kind of information is helpful to people who want to launch collaborations in this direction. So let me just take a pause. It looks like we have a number of other folks who've joined the call and are now promoted up to panelist status. A lot of familiar faces here: Xing, Sulin, and Huifang, and Xing Shu, and Nanshu Lu, Moonki, Junsu. Welcome everyone to this discussion. So I think you just joined recently, so this is just an informal I guess, conversation with Dae-Hyeong, and kind of an interview as well. And so I've asked a few questions. Maybe I can take a pause and see if any of the other panelists would like to ask Dae-Hyeong questions of their own. And if not, I have a long list of questions of my own. We can come back to that. But I'll take a quick break. Does anybody have questions for Dae-Hyeong?
I have a question.
Go ahead.
So, right, I just heard, of course, I always have that impression you're the brightest student from this great man's lab. You must be under tremendous pressure in the early days to try to say, who am I? How can I differentiate with another great man to become a great man myself? How do you, have you asked these questions? Any answers?
First of all, I'm not the greatest student from his. There are so many. Currently, Nanshu, Singh, and Sheng, and Sangmin, and Yang Chai, and so all such, and Hui Fang, and all such previous group members were much greater than me. So I became very humble. But I think that all the achievements were based on close collaboration with my group members. I always worked with a group member, so all the papers that I wrote had 3 equally contributed authors. So that means that the helps from my friends, colleagues, were tremendous and really valuable. So I personally appreciate that help a lot. That's the key.
You're asking the wrong question. You should be asking me how I deal with the pressure because of Dae-Hyeong's reverse. Opposite.
Okay. That question will be reserved for another occasion.
Okay. Actually, John,
yeah.
John gave me a lot of freedom, and so we could be very creative. We could do anything that we want, even something weird thing. He allowed us to do many creative works.
Yeah, Shen has a question.
Yeah, hey, good morning, everyone. Good to see everyone again. So I have a question for Dae-Hyeong. How do you evaluate the impact of maybe one project? Maybe the citation is one metric, or the impact factor, or some other, metrics you have in mind. And once you have those evaluation in your mind, in your own group, sometimes it's like when you publish, you have a lot of impact. Sometimes it does not have as much as you expected. How do you calibrate your system?
I personally don't know. It is quite difficult question. Impact factor is a good good standard to maybe quantitatively evaluate the project. But I think that contribution to the field is important. So new invention and new discovery that can contribute to the field, society. For example, our flexible and stretchable electronic society, and also enlight new young researchers and present a new direction for their future research. That kind of researches can be evaluated as a good research, I think. So I usually talk to my students to try to make new inventions. Since we belong to engineering school, we want to make new materials or new devices. So it's a kind of new invention. But at the same time, we also dig into natural science, especially medical science in biology, and interface between the material and the tissues, things like that. In such cases, we are also seek new discoveries in nanoscale material and biology.
Yeah, to follow up on your comment on collaborating with school of medicine, and also John, I think also asked you this aspect. How do you find the challenge, or how do you deal with the challenges with collaborating in school of medicine doctors?
I have good friends. Yeah, my collaborators are very active, passionate. So they bring their own unmet needs in their field, such as brain science or cardiac applications. But at the same time, they frequently participate in the international conference in cardiology or brain cancer, things like that, and collect some new information and bring it to us for further discussion. So I appreciate to my friends and collaborators in clinical field.
Thank you.
Yeah, just to follow up real quick on that, Dae-Hyeong, how much time and effort do you spend on technology translation? I know you have some funded collaborations with industry groups there in Korea. Is that an important part of your overall set of programs, or do you view that kind of activity as outside of the scope of your university group and you leave that to others? How do you approach that?
Yes, clinical translation is a future goal of our group's research, actually. Since we are working on biomedical devices focusing on brain diseases and heart diseases, we want to transfer our technology to the clinical field. And unfortunately, we cannot perform industrial projects because our funding, IBS funding, doesn't allow to have the industrial funding. But we are very much interested in the clinical translation. And for example, today I will talk about cardiac device that can treat arrhythmia, myocardial infarction, and the heart failure, and also biodegradable devices for the treatment of the brain tumor. Both are very, very challenging diseases, and there are a number of patients are dying right now because of those diseases. So the motivation given to my students is very strong. And although it will take very long time, I guess that clinical translation needs both lab scale experiments and large animal scale experiments, and eventually human trials. And even after that, we need approval from the FDA. So all procedure, each step is very, very challenging and takes long time. So we hope to be patient to get through that such long journey. But yeah, anyway, that is our ultimate goal and very, very much important.
Yeah, great. Thanks. That makes sense. So it looks like we have a couple of hands raised here. Maybe we'll start with Nanshu Liu. Hi, Nanshu.
Hello, John. Hello, Dae-Hyeong.
Hi, Nanshu.
Yeah, everyone. Good morning, good evening to everyone. It reminds me of our early morning 7 a.m., 8 a.m. group meeting. Now it's the third generations. So this is truly exciting, and thanks EML for inviting Dae-Hyeong, of course. So Dae-Hyeong, you mentioned that a lot of your papers have like 2, 3 equal first authors. Seems like you have a really generous heart. You really give your collaborators and your students nowadays a lot of credits. And it's tremendously important, of course, for young scholars and scientists. So, but we also know not everybody is like this. A lot of people fight for credits and eventually have damaged the relationship. So you start to understand the importance of collaboration and sharing, like when you are very young. And I'm just curious, where did this generous heart come from, and what is the philosophy that supported this kind of sharing?
First of all, I really deeply thank to my collaborators for their excellent contribution, including you. So without Nanshu, probably I couldn't do so great works when I was in Illinois. So it's not because of the generosity, it's natural. Since your collaboration was so great, I had to do that. But I hope to teach my students to be generous to each other, to share knowledges and to share credits. And actually that can accelerate the entire speed of the research, because they can focus on their expertise, and they can get helps from other people who have different expertise. So the entire progress of the research can be accelerated, and so that is beneficial to both researchers, I think. Thank you.
Yeah, I think that's a really important thought, Dae-Hyeong, and you've done a great job with that. I know mentorship, it's an important part of being a professor. I know you are very passionate about, that role, that you play in your group. Do you have some other thoughts on your philosophy around advising your PhD students?
Yeah, I think that frequent discussions between me and students, as well as between students, group members, are important. So talking to each other frequently, rather than focusing on their own project only, is important. So I try to have two kinds of team meetings, two kinds of meetings. One thing is a team meeting, the other thing is a private meeting with my students. So during the team meeting, active collaborations between lab members can be made, and students can share their knowledges and experiences each other, and that should be very beneficial to each group member. At the same time, I am trying to have frequent private meeting, just me and my student. So in that case, we can see more details deeply. And any difficulties in the experiment and the planning and future research direction and the career paths. So all such detailed private things can be discussed with me. So yeah, such discussions seems to be important.
Yeah, great. That makes a lot of sense. So it looks like Junsu Kim has a question. So you want to go ahead?
Yeah, hi all, I'm Junsu, fourth year graduate student in Jiang's group. And actually, I met Professor Dae-Hyeong Kim before I go study abroad, and he gave a very valuable comments. I remember that moment. And yeah, most of my questions were already answered, but I have one more question. So in terms of the culture, sometimes students are afraid of professors because of the some hierarchy, or sometimes students cannot say opposite opinion in front of the professor. Right after I came America, I felt very big difference in culture. Right now, if I don't like Jigang's opinion, I can say, oh, I don't like this idea. Maybe it is wrong. So in Korea, I remember most of my friends were afraid of saying no in front of professor. It seems you are not. How do you deal with this kind of open? How did you make this open discussion culture in your lab? How, yeah.
I'm not sure I'm good at it. Yeah, I agree with that. Jigang and John are have very open-minded, and to them we can say no. But yeah, I'm not sure if my students can feel in the same way to me. But at least I want to try to be open-minded to my students, and so that's the reason why I have periodic, frequent private meeting with my student in my office. We can sit together closely, and we can just spend 30 minutes. Although we do not have many things to discuss, we can just sit and chat. Then maybe more friendly mood can be made, and they can say anything that they want during that period. Yeah, but I'm not sure I'm very good at that. Yeah, in comparison to Jigang and John, they are really nice.
Yeah, that's a very good approach, Dae-Hyeong. It looks like Wei Fang has his hand raised, so welcome, Wei.
Thanks, John. So Jigang asked about the time you're in John's group and the pressure. I have a follow-up question about after John's group. We are all trainees from men like this, and then casting such a big shadow of kind of scientific research. So how do you deal with the fact that there's such big shadow? How do you successfully identify yourself? I see that I feel that you've done it very well, but I see that it's a big challenge for myself, at least. Whenever I want to do something new and I do little research, I see John has published it a long time ago. So
that's true.
I feel probably there are similar challenges from Jigang and Jimmy's trainees.
That is really challenging, since yeah, I admire Jigang's work and John's work very much, and also it is also true that their research always inspires us for the next step and the next direction. But I remember that John gave me a comment when I left his group that I should be independent rather than relying on him. So that is an important criteria that I have to think of when I become an independent principal investigator. So yeah, I tried, but although it was challenging, I tried to be different. And yeah, it is true that doing completely different research from John Rudder's group is really challenging. I have to admit that. But in details, I think that we can differentiate things, especially in the case of biomedical device research. There are various kinds of specific disease models, and even in brain science or cardiac research, if we go into details, then all situations are different, and maybe we want to optimize materials and device shape to be specific to that detailed application, biomedical application. So yeah, that and also that comment was given by Nanshu when we discussed about the new research direction. Nanshu gave me the comment that we can be more specific to the specific disease model. I mean, we can differentiate ourselves from Raja's group by focusing on specific disease model. Yeah, I just followed her guide. That's what I did.
So I have to say I'm detecting a recurrent theme in some of these questions, which is something along the lines of the old guys need to get lost. Too many problems coming from us old folks. Anyway, I think we're almost toward the end of our interview period, but I don't want that to be the parting thought. So let me ask one more question of Dae-Hyeong. What are your career goals 5, 10, 20 years out? Like, what are you hoping to accomplish in those different time frames?
Yeah, I don't know. I'm just trying my best with the current situation. That's what I'm doing. Maybe, but I just want to spend more time with my students. And if I choose one goal, I hope to make a meaningful clinical translation of the bioelectronic device. So I just want to help patients.
Yeah, great answer. So Jimmy, should I hand it over to you at this point? I think
sure.
Do you want to go through?
We'll have a couple of announcements to make. So let me share my screen. And do you all see the slides?
Yes.
Okay, so this is EML Webinar, and EML stands for Extreme Mechanics Matters. And we just got our new 2020 impact factor of 4.567. I don't know how to calculate that, but we could have done better than this. This is the team EML. 3 of us, John Rogers, Zhigang Su, and myself, are editors-in-chief. We have 5 editors at the bottom, and we have publisher Rebecca Capun from Elsevier. I just want to take the opportunity to promote a few recent papers. Madden City wrote a perspective on physical intelligence as a new paradigm. Fascinating paper. Go and read it. It's published in EML in 2021. This is the era of COVID-19. Tahir Saif and his colleagues, students, actually published a paper about how to measure the effectiveness of masks using fluorescent to tag the particles or droplets and tracing that using fluorescent microscope. And there's another paper published in 2021 by a Dutch group on mechanical binary memory device. They called it monolithic binary stiffness building blocks for mechanical digital machines. Go and read them. Zhigang, you want to say a few words about how we started?
Oh, EML webinar, this has been repeated so many times, started when in the early days pandemic in the United States. And John and I and Jimmy suddenly found us a lot of time to do, no students to abuse. So so we heard maybe this is a way to spend time together online. So at that time, really, we didn't have Zoom fatigue, just didn't know what to do with Zoom. So EML webinar started. We have one webinar every week until everybody is tired. In the first season, we have a total of 32 webinars. And one outstanding feature of EML webinar is extensive discussion. Each webinar is about 3 hours, a little over 3 hours. You can do calculation yourself. So there's over 100 hours of videotapes all on Google on Google, YouTube. an enormous number of views. This is not the most current number. The current number must be over 100,000 views. Thank you.
Okay, and so based on that success, we started season 2 for some reason. Okay, this is EML Webinar season 2, and we decided to reduce the frequency, but emphasize interdisciplinarity. And we also introduced some innovations. For example, we introduced the WeChat live streaming in addition to YouTube live streaming. For the first webinar of this particular season, we actually had more than 12,000 viewers from WeChat live streaming. And here are a few webinars that we have had for season 2, and 2 of them are actually providing EML webinar overviews. This is another feature. So people can go back and watch the webinars, but we also publish the overviews. So Michael Sheets and Julie, each of them would publish an overview in EML about their webinar. Okay, let me hand it over to Teng. So we are also doing some social experiments.
Thank you, Jimmy. And before I start this this slide, just want to let Dae-Hyeong and John and all the panelists and all our viewers know that while we are talking right now, in the WeChat live stream, there are already more than 3,000 viewers of this EML webinar right now. Even before we started the official webinar by Dae-Hyeong, just the first half an hour interview was very intriguing. And in this season, we start the fundraising as we need some very basic fundings to keep EML webinar going and to have the independent Zoom service, cloud storage, and so on. And so far, we have raised $1,715 from the donors listed in below, and we would like everyone to consider to donate to keep the EML webinar season 2 going. If you can do a screencast of the QR code on the screen and take out the smartphone and scan, you can donate while we get started for today's webinar.
Thank you very much, Teng. That's it. So let me get out of this, stop sharing. We'll go back to the Dae-Hyeong's webinar.
Okay, great. Thanks, Jimmy. So Dae-Hyeong, if you want to go ahead and share your screen, I'll say a few words of introduction and then it'll be all yours and we'll listen to your seminar. So it gives me great pleasure to introduce Dae-Hyeong Kim. He's on the faculty at Seoul National University. He'll talk to us about unconventional bioelectronics for heart and brain diseases. Let me say a few words about Dae-Hyeong's background. So he got his undergraduate degree at Seoul National University in chemical engineering. And looking at his CV, I didn't realize he has a minor in business administration. So he's kind of a business guy. I didn't realize that. He then got a Master of Science in Chemical Engineering, 2002, also from Seoul National University. And then he went to industry. So he worked for a company called KC Tech. I think it was a photoresist company, some kind of semiconductor materials processing related company. But he was there for 4 years, from 2002 to 2006. Then he came to University of Illinois, Urbana-Champaign, and I was lucky enough, I kind of hit the jackpot when he decided to join my group. Had a spectacular run as a PhD student, and then as a short time as a postdoc in my group, I think finishing up in 2009, that sounds about right. And so he joined the faculty at Seoul National University immediately after that. He's grown a large and productive group almost instantaneously. It's amazing the trajectory that he was able to follow, as an independent researcher. But He's won many awards. He won the gold medal, the MRS Graduate Student Award in 2009. He was selected by MIT's Technology Review magazine to the TR35 group in 2011. He's a regular, highly cited researcher by Clarivate Analytics almost every year these days. He's now associate director as well of the Institute for Basic Science at Seoul National, and so he's held that position since 2017. And so he's just been putting out an amazing collection, a steady stream of really interesting research results out of his group. And I think he'll share some of the latest with us in his presentation. So it's all yours, Dae-Hyeong.
Thank you very much for the nice introduction. And also thank you again for the kind invitation. Today, I will talk about unconventional bioelectronics for heart and brain diseases. This schematic illustration summarizes our group's research. We have been working on nanomaterials-based soft bioelectronics, such as soft-type implantable devices as well as wearable devices. And today, I will focus on two topics, devices for heart disease and the brain disease. So in the first part, I will talk about cardiac mesh and cell sheet transfer printing. And in the second part, I will talk about soft devices for glioblastoma treatment and fatal seizure treatment. Let me begin with statistics. This chart shows the number of deaths in between 1969 and 2020. And as you can see, heart disease has been the #1 cause of death historically. In another statistics, more recent one, again, heart disease is the #1 cause of death. So it is very important to understand heart disease and develop appropriate device to treat heart disease properly. And if we look into the cardiac deaths, the statistics of the cardiac deaths, then more than 50% of cardiac deaths is caused by coronary heart diseases, such as myocardial infarction, arrhythmia, and heart failure. So naturally, our focus was on the development of devices for coronary heart diseases such as MI, arrhythmia, and heart failure. And before going into more details, let me briefly explain the development process of the heart disease. Usually it begins with the narrowing of the coronary artery and due to the accumulation of plaques inside the coronary artery, blood supply will be reduced to these cardiac muscles. So cardiomyocytes will become dead, and which leads to myocardial infarction and arrhythmia, and eventually a severe remodeling of the heart shape will occur and leads to the failure of the heart pumping function. And we have been working on these three cases. For example, back in early 2010, when I was in Illinois, we developed a multifunctional balloon catheter. In this case, we designed stretchable electronics, islands interconnected by serpentine-shaped interconnection, and stretchable electrodes and pressure sensors were mounted on the inflatable balloon catheter like this. So this balloon catheter was inserted into the blood vessel and reached the heart chamber and made a conformal contact to the endocardial surface to measure electrocardiogram and the pressure changes. In another example, again, back in 2010, we worked on the epicardial mapping array for the detection of the onset of arrhythmia in collaboration with the researchers at UPenn. In this case, we used ultra-thin doped silicon nanomembrane to fabricate multiplexed electrode array. Since we used very thin 300 nanometer thick silicon nanomembrane, the flexural rigidity of the silicon could be reduced dramatically. So the entire multiplexed array could be very much flexible. So it could make very good conformal contact to this swine hot surface and could measure the propagation of the electrocardiogram with high resolution. Actually, this was the first work in the bioelectronic device in which we applied a silicon nanomembrane. After I came back to Korea, our focus was turned into heart failure, and this movie explains the transition from healthy heart to heart failure heart. In the case of a healthy heart, usually the shape of left ventricle and the right ventricle are symmetrical, and the beating of LV and RV are symmetrical. So the pumping function, pumping capability is high enough. But in the case of heart failure heart, first of all, the shape is asymmetrical, and the beating of cardiomyocytes in LV and RV are not synchronized. So due to that non-synchronized beating of cardiac muscles, the pumping capability can be decreased a lot, which eventually lead to heart failure. So to treat that situation, electrical stimulation therapy as well as mechanical passive ventricular restraint approach have been used or developed. In the case of electrical stimulation therapy, typically a device is implanted around in the chest, and electrodes are connected to the implanted device, and these electrodes are inserted into the blood vessel to reach left ventricle and the right ventricle, and these two electrodes stimulate LV and RV simultaneously in order to make the LV and RV beat at the same time. And theoretically, it should improve the pumping function a lot, but in reality, the efficacy is limited, all because of the localized beating, localized stimulation. In other words, although we apply electrical stimulation in this point, since already cardiomyocytes and cardiac muscles are dead due to the blocked coronary artery, and typically the dead cardiomyocytes show delayed conduction. So the electrical stimulation signal cannot be spread over the entire ventricle properly. So the non-synchronized beating still occur. And the solution actually is to insert more electrodes. Maybe if we can use a number of electrodes that can be positioned in these positions, then we can apply the electrical stimulation signal to entire ventricle, so make all the alive cardiomyocytes beat at the same time, contract at the same time, so we can maximize the pumping function. However, unfortunately, this is not possible. First of all, we cannot insert tens of electrodes into this narrow blood vessel. More importantly, these electrodes should pass through valve, cardiac valves. And to be these electrodes are supposed to be located over a large area of the ventricle, which is not feasible because there should be side effects in the valves. So instead of using this endocardial approach, we need another approach, epicardial approach, to solve this issue. In another approach, mechanical approach, a rubber-based cardiac mesh can be used. This elastic device encircles the heart and share myocardium stress by applying compressive force to ventricle. So it can apply pressure when ventricle contracts, so it can increase the blood pumping pressure. Although beneficial, the efficacy is limited because it is not involved with the electrical stimulation. So our approach is to combine these two. So we fabricated a cardiac mesh whose shape is quite similar with this one. And also this device is elastic because it is based on rubber. But instead of using conventional insulative rubber, we try to use conductive rubber. then the electrical stimulation signal can be applied over entire ventricle and like this. So we can use mechanical benefit as well as large area global stimulation advantages by using this device. But although the concept is unique, we had issues in materials. For your information, the typical conductivity of the electrode, the metal electrode, is higher than 100,000 Siemens centimeter, although the commercial conductive rubbers show the conductivity way less than 100. There is a big difference, and because of this low conductivity, in other words, high resistivity, high power consumption, as well as delayed signal conduction can occur. So the first thing that we had to do is to develop a high conductivity rubber first to fabricate this device. So we take a look at the conductive rubber, and typically conductive rubber consists of two components: elastic rubber matrix and filled with the conductive fillers. And in the case of conductive fillers, there are several candidates, such as carbon-based materials such as CNT or graphene, conducting polymers or metal-based nanomaterials. And as you can see, the silver-based nanomaterials show the best conductivity among all materials. And in particular, if we can use ultra-long silver nanowires, then we can minimize the percolation threshold of the conductive network. In other words, with a small number of nanowires, we can make a conductive pathway of the nanowire network. So we chose ultra-long silver nanowires as a conductive filler. And in the case of elastic media, we can use either chemically cross-linked polymers such as PDMS or Ecoplex, or thermoplastic polymers such as the SBS or SCBS. In the case of PDMS, for example, the chemical stability is very good. However, to cross-link PDMS, then we have to mix prepolymer with a platinum-based catalyst for polymerization. But unfortunately, if we add a large number of conductive fillers, such as silver nanowires, then platinum catalysts become inactive. So the polymerization cannot be proceeded. So there is a limitation in increasing the loading amount of the conductive filler in the case of chemically cross-linked polymers. So instead, we used thermoplastic polymers. In these cases, as far as we can apply appropriate heat, we can proceed the polymerization, although the loading amount of the conductive fillers is high enough. So by combining, by using the thermoplastic elastomers and ultra-long silver nanowires, we fabricated this cardiac mesh that wraps around the animal heart, which is a post-myocardial infarction model, and restore the normal heartbeat. Although we proved the concept, the efficacy of the concept, still we had many issues toward the clinical translation. In particular, we had many issues in material. For example, silver nanowires can be easily oxidized. And if silver nanowire is oxidized, then the contact resistance between silver nanowires will be increased a lot. So inside the animal body, as time goes, the conductivity will be continuously decreased because of the oxidation of the silver nanowires. Also, inside the biofluids, silver nanowires can be dissolved, and silver ions can be leached out, and such silver ions are toxic. So we have to solve the biocompatibility issue. We also wanted to have higher conductivity, and the easiest way to achieve higher conductivity is put more nanowires into the composite. But as we add more nanowires, then the composite will lose the intrinsic rubber-like property. Rather, it will become stiff and can be broken easily. So there is a trade-off between the conductivity and the softness, although we have to improve both. So to solve these issues, we replace the silver nanowire into cold-shell type nanowire. So we stick with the ultra-long silver nanowire because it provides a very high conductivity and low percolation threshold. But instead of using bare silver nanowire, in this case, we coated the surface seamlessly with gold shell. Then, since gold shell is inert, the oxidation of silver nanowire can be prevented, and also silver nanowire dissolution can be prevented. So we could solve the oxidation issue as well as the biocompatibility issue. In addition, we optimized the polymerization condition, such as the polymerization temperature or additive amount to induce local microscale segregation. So we could achieve high conductivity and stretchability because of that special structure. So this movie shows the stretchability. For information, previously, when we fabricated the silver nanowire-based composite, the stretchability was 50% and the conductivity was 7,000 joules per centimeter. But in this case, the maximum stretchability could be over 800%, and conductivity could be more than 70,000 joules per centimeter. So although still lower than the intrinsic conductivity of silver, bulk silver, but this is a very good improvement in comparison with the previous case as well as commercial conductive rubbers. And we confirmed the biocompatibility through the in vitro experiment as well as in vivo animal experiment. And due to the gold shell coating, we could prevent oxidation of silver nanowire, so could maintain the initial conductivity over the long period of time. And by using this material, we fabricated multi-channel cardiac mesh designed for the peak hut. We first imaged the peak hut and made a model and customized the design of the multi-channel cardiac mesh as shown in this image. Since we were interested in the local EKG recording as well as the local electrical stimulation, we paired two electrodes into one channel. And through these channels, we could measure the electrocardiogram on various points of the peak hut and could make an activation map as shown in this image. And based on this activation map, we could figure out the damaged part of the heart ventricle, which shows the late activation. And based on that scarred region, we customized the electrical stimulation protocol to make all the alive cardiomyocytes beat synchronously and maximize the pumping capability. That is the first part of the cardiac about the cardiac mesh. And although the cardiac mesh could maximize the pumping function at the specific status, but it cannot regenerate the dead cardiomyocytes. So there has been another effort using cell sheet transfer toward the regeneration of the dead cardiomyocytes. The cell sheet transfer therapy was presented by Taro Kano group in Japan. They cultured the cell sheet on the polypalm substrate. And such a cell sheets can be either cardiomyocytes or the mesenchymal stem cell. And if the temperature is changed from the body temperature to lower temperature, such as 20 degrees Celsius, then the surface property of the PPAM layer is changed. So due to that changed surface property, the cell sheet can be delaminated and can be transfer printed on the target region of the heart. And later, in 2017, another group in Japan used this transfer printing technique to try the clinical trial of the stem cell sheet transplantation therapy for treating cardiomyopathy. And according to their conclusion, cell sheet transplantation was feasible to treat cardiomyopathy, and they confirmed this treatment's efficacy for severe congestive heart failure. So the feasibility of this cell sheet therapy was proved. However, the challenge is in the temperature control, precise temperature control in the clinical field. Actually, we need to transfer several cell sheets to cover large area of a human heart, a damaged part of the human heart, as well as to transfer print multiple cell sheets onto the damaged cardiac tissue. That means we have to control the temperature multiple times during the clinical procedures and surgery, which is quite challenging. So we tried to develop another type transfer printing method by using PDMS stamp. We have been working on transfer printing of various kinds of nanomaterials, including silicon nanomembranes using PDMS stamp. Typically, we use the kinetic control. But in the case of cell sheet, we modified such transfer printing a little bit. First of all, we have to culture cell sheet on the PDMS. If you try culture cell sheet on the bare PDMS, it should be very difficult because cell sheet cannot adhere to the PDMS surface because PDMS has very low surface energy. So in our case, we laminated a very thin graphene layer on the PDMS surface. Since the graphene layer is very thin, it doesn't affect the soft nature of the PDMS stem. And graphene has very good adhesion with the cells, such as cardiomyocytes or mesenchymal stem cells, so we could culture cell sheet on the graphene layer. In addition, we patterned the graphene layer as a lioness base for the purpose of alignment of cell sheet. So this is a magnified view of the cultured cell sheet, which shows the good alignment of molecules. And after culturing the cell sheet, now we need to transfer print. And our transfer printing process doesn't require temperature change. Rather, we need a two-step procedure, preparation step and the transfer step. What we need to do is just to contact the cell sheet to temporary plastic substrate and detach it. That's it. If we make a conformal contact of the cell sheet to polystyrene substrate, for example, and detach it, then crack will be generated at the edge of the cell sheet. Then this cell sheet is now ready for transfer printing. So after generating crack, if we make a contact of the cell sheet to target substrate such as protein-coated glass or on the target region of the heart, the cell sheet will make a good contact to the target substrate. And if we gently remove the PDMS stamp, then the cell sheet can be simply transfer printed as shown in this image and large areas of cell sheet transfer printing image. Also, we can repeat this transfer printing process several times to make a stack of 3 layers of cell sheet or 5 layers of cell sheet. That is beneficial to improve the efficacy. As we increase the thickness of transfer cell sheet, the therapeutic efficacy, including paracrine effect, can be improved. And this movie shows the crack generation, the magnified view of the edge part of the cell sheet. So after making a contact of the cell sheet to the plastic substrate, and if we detach it, then crack will be generated at the edge of the cell sheet So the cell sheet will be shrinked a little bit, and we can see the crack generation through this movie. And then the cell sheet is ready for transfer printing, and then we can make a conformal contact to the target substrate. In this case, we used protein-coated glass. Actually, transfer printing yield to the animal tissue is higher than the glass substrate because typically animal tissue has many amine group, and so good covalent bonding and hydrogen bondings can be made between the cell sheet and the tissue. After making a contact, we can wait a little bit, roughly 90 seconds, and peel the PDMS stamp. Then we can transfer print cell sheet. Since cell sheet is transparent, it's a little bit difficult to see, but if you see the reflection, then the cell sheet was transfer printed to the glass substrate. So now, and that process can be repeated several times. This movie shows the confocal microscopy observation of the stacked cell sheet. So the bottom and top layer was stained with red dye, and the middle layer was stained with the green dye. And by changing the focus of the confocal microscopy, we could confirm that a stacked cell sheet could be prepared properly. So now we are ready for transfer printing of cell sheet for cell sheet therapy. But there is another step that we can think of before widespread wide application of the cell sheet for medical field. We need to culture a large number of cell sheets. First of all, if we think about the animal heart is very small, so maybe one cell sheet is good enough, but the human heart is much larger than the mouse heart. So to cover large area, we need to transfer print in multiple cell sheets. In addition, if we can stack multiple cell sheets, usually the therapeutic efficacy is increased. So typically we stack 5 layers or 10 layers of cell sheet. That means we need to transfer print multiple layers of cell sheet. Actually, that initiated the development of the new transfer printing method, because it is quite challenging to change the temperature between body temperature and low temperature several times. Rather, this process, transfer printing process, is much easier. But anyway, that means we need to develop a method to culture cell sheet in large quantity. And this movie shows the industry level incubator for large scale cell sheet culture. The employee needs to wear this kind of costume, special costume, to prevent contamination. And periodically, the personnel need to take cell sheet out of the incubator and stain with a special dye and observe the cell culture process, such as proliferation or differentiation through the microscopy for the quality control of the cell culture process. And the entire process is very labor intensive, very expensive procedure. And also, whenever the incubator is open, then the cell sheets are taken out of the incubator. There is a contamination risk, and CO2 environment inside the incubator can be broken. So it's not very ideal. So we thought that we can work on the development of novel mass cell sheet culture process by using our technology. So our goal is to change the conventional labor-intensive cell culture process into digitalized mass cell culture process, which can minimize the manpower and the contamination risk. As shown in this illustration, in the typical mass cell culture process, many people need to work on the quality control process, and that involves with contamination risk. But if we can develop a special sensor and actuator which can be put in the incubator and collect the data wirelessly and send a feedback simulation signal wirelessly, then maybe one person can control the entire cell culture process and entire incubators. So to achieve that goal, we designed and fabricated this wirelessly integrated mass cell culture platform, as shown in this image. The pink color corresponds to the cell culture media, and we have here 5 layers of cell culture substrates. This is the image of the 5-layer cell culture substrate. Each substrate was fabricated by using 3D printer, and we transfer printed ultra thin sensor and actuator array on the substrate. And on top of this sensor and actuator array, we can culture cell sheet, so 5-layer cell sheets. We can also increase the number of layers up to 25 layers as far as we can use the automatic media circulation. If we can use this kind of media circulation, we can maintain the cell viability over 90%, although we have 25 layers. But in this case, anyway, we just use the 5 layers, and each sensor and actuator array was connected to the Arduino board through the ACF cable. And on each substrate, we have multiple impedance sensors, temperature sensors, potassium sensors, and pH sensors, as shown in these images. And these sensing data could be collected wirelessly through Arduino board, and all the data can be collected in the central server. So the one person can analyze all the data collected from each cell culture substrate. Also, if a specific abnormal signal is detected, then the personnel can control the cell culture platform through wireless communication. For example, electrical stimulation or thermal stimulation can be applied to specific position of the cell culture substrate, or the medium exchange and the temperature of the incubator can be controlled wirelessly. So the entire monitoring and the control procedures can be done through the wireless system. And this slide shows the characterization result of each sensor, pH, potassium impedance and temperature sensor, and the electrical and thermal stimulation, and the selectivity of the ion sensors. And each cell culture platform can be stacked like a Lego block and put in the incubator. And after closing the incubator, we can collect the sensing data wirelessly. And this shows the user interface programmed by LabVIEW by my students. And after analyzing the data, we can send a specific control signal to the cell culture platform through a wireless system, and control the cell culture medium circulation and electrical stimulation and thermal stimulation. And this shows the monitoring data and the stimulation result. We cultured 2 types of cells, such as mesenchymal stem cell or cardiac muscle cell. In the case of mesenchymal stem cell, after seeding the cell will be cultured, and when 100% confluency was achieved, the impedance becomes saturated, so we can see the end of the proliferation. And in the case of cardiac muscle, during the proliferation process, impedance continuously increases, but after that, differentiation will begin and conducting myotubes will be formed. And because of the conducting myotube formation, the impedance will be decreases slowly. So we can follow up the entire proliferation and differentiation process. And all these impedance data could be collected from 16 different positions on one substrate, and each impedance data can be collected to make a three-dimensional map as shown in this image. Also, the temperature data and the pH data and the ion concentration data can be collected also. And if there is any abnormal signal or situation are observed, then we can control the circulation system or the temperature, or we can apply electrical thermal stimulation through wireless communication. And in more detail about the stimulation, if we apply electrical or thermal stimulation, then we can accelerate the proliferation process as also differentiation process. For example, if we want to accelerate the cell culture process only third layer or these 2 points, we can apply specifically these 2 points or this layer, then we can make the cell culture process much faster than the other positions. So we could demonstrate that we could control the cell culture process through this system. And we expect that using this system, the entire cost for the mass cell culture process can be reduced dramatically, and that will facilitate the widespread application of the cell sheet therapy in medical field. Actually, that mass cell culture business is very active in Europe in the cosmetic industry. So in Europe, the animal experiment for toxicity of the new cosmetic products is banned right now. So instead of using animals, this kind of cell sheets are being used by the cosmetic companies, and that business is done by Episkin, which is a subsidiary company of the L'Oreal, and they are using mass cell sheet culture process, and hopefully our technology may be helpful for that kind of industry. That's the end of the cardiac part, and let me move on to the brain disease part. Again, this chart is showing the number of deaths, and historically, as I mentioned, heart disease has been the #1 cause of death. But if we look at the increasing trend of the cancer deaths, sooner or later, cancer deaths can be the #1 cause of death. And especially the malignant tumor is very troublesome. That increases the cancer deaths a lot. And there are several malignant tumors whose 5-year survival rate is very low. Especially the brain tumor is very fatal. And more serious issue is that in past 40 years, the improvement of the 5-year survival rate of the brain tumor is very marginal, much less than other types of tumors. And therefore, the 5-year survival rate of glioblastoma is still 10%. That means 90% of patients will die after 5 years. That is quite a fatal disease. Now, why is glioblastoma so fatal? Is related with the special feature of the brain. In the case of other types of cancers, typically first we remove the tumor tissue through surgical resection, as well as surrounding tissues over large area. Surrounding tissues will be removed surgically. And such surrounding tissues may contain some tumor cells and may induce recurrence of the cancer. But since in other types of cancer, surgically, the surrounding tissues are also removed, so the tumor recurrence can be suppressed. But in the case of brain, that strategy cannot be applied. if we remove large part of the brain, the patient will lose certain brain function permanently. So typically, rather than large area surgical resection, only the tumor, the core part of tumor is removed. So incomplete resection will be resulted in due to the feature of the brain. And some margin should remain. And of course, that marginal region contain tumor cells, and that can cause the recurrence of the brain cancer. So typically, to prevent the recurrence, radiotherapy and chemotherapy are followed. But due to the radiation tolerance, the radiotherapy has a limitation. So our hope is in chemotherapy. And in fact, in other types of cancers, chemotherapy is a very powerful tool to suppress the recurrence of cancer tumor. But in case of brain tumor, this doesn't work properly because of the BBB. BBB, blood-brain barrier, is a natural barrier that protects a brain. Since brain is so important organ in our body, BBB hinders the access of external organism to the brain tissue. But unfortunately, that BBB also blocks the delivery of the chemo drug. So due to the incomplete, inefficient delivery of the chemo drug from the bloodstream to the brain tissue, the efficacy of the chemotherapy is quite low. So this is all about the delivery issue. That means instead of using systemic delivery, if we can develop a new localized drug delivery method, then maybe we can improve the efficacy of chemotherapy and contribute for the brain tumor patients. And that idea was taken by researchers at Johns Hopkins already. They developed a biodegradable device that can be implanted inside the brain cavity, which is called as a gliadar wafer. This looks like a chip and rigid. So a biodegradable polymer consists of biodegradable polymer and chemo drug called camostin. And this chip, gliadar wafer, can be implanted in the brain cavity. So after removing the brain tumor, there should be a cavity, and inside the cavity, this gliadar wafer can be implanted. And after skirt is covered, the CSF, cerebrospinal fluid, will fill this cavity. So the gliadar wafer will be dissolved, and the loaded drug will be released. It takes about a week to release entire drug. And because of that 1 week intracranial drug delivery, the 2-year survival rate could be improved up to 20%, much better efficacy than the other approaches. So this gliadar wafer was approved by FDA and has been used for the treatment of glioblastoma in the United States, and could be very helpful to help patients. But unfortunately, still the survival rate is low. 80% of people can die after 2 years. So maybe we can add a new device, I mean the biodegradable electronics, to this biodegradable wafer to control the drug delivery process more efficiently, and to deliver drug more efficiently, and eventually to improve the survival rate. And I'm sorry. And we were inspired by this amazing work, transient electronics. In this case, a silk substrate was used as a biodegradable substrate, and on top of that, we took in silicon nanomembrane, and also magnesium electrodes were used to fabricate biodegradable electronics and bioresorbable electronics, of course. And inspired by this work, we fabricated a flexible, sticky, biodegradable, and wirelessly controlled drug delivery patch. In this case, we used oxidized starch. As you may know, a starch is biodegradable polymer, and if we include oxidized units in starch, it becomes very sticky. So it can show very good adhesion to the tissue. This can make a covalent bonding with the amine group of the tissue and the hydrogen bonding. So good adhesion can be made. In addition, doxorubicin, a representative and famous chemo drug, can be covalently conjugated with the oxidized starch polymer chain. Since doxorubicin has red color, so the drug reservoir has red color. Drug reservoir is made of oxidized starch containing doxorubicin. And separately, we fabricated biodegradable devices such as wireless heater and the temperature sensor based on magnesium, and that devices were transfer printed on the drug reservoir. And after surgical resection of the brain tumor, this patch could be implanted into the surgical cavity. Since the device has a thin thickness, is flexible, so it can make good conformal contact to the curvilinear brain cavity surface. And due to the oxidized unit, it is sticky and make a good adhesion. And the device is biodegradable. And after covering the skull, we can apply magnetic field, and that magnetic field can induce eddy currents in the magnesium-based heater, and that eddy current induce Joule heating and make local thermal actuation, and that local heating can increase the doxorubicin release amount, as well as the penetration depth of the doxorubicin molecules into the brain tissue, which is beneficial for the treatment of the remaining GBM glioblastoma cells. Let me explain more details. We started with the cone powder. We oxidized the cone powder and mixed with the doxorubicin. Then doxorubicin, some doxorubicin molecules can make covalent bonding conjugation, while some doxorubicin molecules are still just physically trapped inside the polymer chain. And when we evaporate the water, then we will be able to obtain, we obtained this OST dox patch. OST means oxidized starch, and dox means doxorubicin. And we can, another type of course, another type of chemo drug, such as temozolomide, which has been used for the GBM treatment. But temozolomide doesn't have a color, and since we wanted to observe the diffusion of drug into the brain tissue, we used the doxorubicin for our experiment. Of course, we can use both chemo drugs in one patch to cope with the heterogeneity of the glioblastoma. And after making the drug reservoir, we separately fabricated the device, magnesium-based device, and transfer print those devices onto the patch and cut the patch. And after fabricating this device, we packaged it and sterilized with the ethylene oxide gas. Note that we cannot use conventional autoclaving because hot steam may dissolve these magnesium-based devices. So instead of using hot steam, we used ethylene oxide gas for sterilization. And the entire device is biodegradable. The drug reservoir is made of OST. Encapsulation layer was made of PLA. The device was made of magnesium. So after hydrolysis inside the brain, the byproducts were resolvable, and we confirmed the biodegradation in vivo inside the animal brain. And after 10 weeks, the entire device could be dissolved and resolved by the body. And the device was thin enough to be flexible and make a good conformal contact. This image shows the magnified view of the interface between the patch and the device. We can see the very good conformal contact. Also, since we included oxidized units in starch film, it could make a good adhesion to the brain tissue. Also, it could make conjugation with the doxorubicin molecules, while some doxorubicin molecules still are physically trapped only. And that conjugation is helpful for long-term sustained drug delivery. For example, if we do not use the oxidization, then most of doxorubicin molecules will be just physically trapped. And these physically trapped drug molecules can be released easily. For example, within 1 or 2 days, almost 80% of loaded drug will be just released if we do not use OSD film. But if we use oxidized units, oxidized starch film, then due to the conjugation, sustained long-term drug delivery can be achieved. According to many literatures in cancer treatment, rather than intense burst release of the drug, long-term controlled drug delivery is much more beneficial for the treatment of the cancer. So this is the more ideal than this kind of burst release. Also, the cavity is filled with CSF, cerebrospinal fluid, and without the encapsulation layer, the loaded drug will be released to CSF. Then we will waste the valuable loaded drug. So we passivate the surface with hydrophobic PLA layer to prevent unwanted drug leakage to CSF. So after encapsulation, we could suppress the drug release significantly. After implantation, we can apply external magnetic field, typically 30 minutes per 2 days. And if we apply the magnetic field due to Joule heating, the drug release will be accelerated from 25% up to 75%. And also drug penetration depth can be increased a lot. So without heating, the penetration depth is shallow. However, with heating of 5 degrees Celsius, the penetration depth can be increased much. We can control the temperature increase below 10 degrees Celsius, typically around 5 degrees Celsius, and that could be measured by using the temperature sensor. And we apply this system to animal model. We first grow the GBM cells. We use the U87MG cell line, GBM cell line in biopsy nude mouse, immune deficiency mouse. And after cell growth, tumor growth, we surgically remove the core part of the tumor and implanted the device in the cavity. And for 2 weeks, we applied 30 minutes wireless actuation, thermal activation for 2 days, every 2 days. And after 2 weeks, we just waited the biodegradation of the natural biodegradation of the implanted drug, so implanted device. So we waited for natural release of the drug due to the biodegradation. And according to the tumor data, so after 4-week treatment, the tumor volume could be dramatically suppressed when we use the OSD film with the wireless heating. The tumor volume was way smaller than the control groups, and that suppressed the tumor volume agreed with the survival rate. We followed the survivor rate about 70 days. And in the experimental group, the survival rate was over 80%, while in control groups, the survival rate was much lower. But still, we have 20% decrease, and that means that tumor regrowth can occur. So more recently, we developed a second follow-up treatment method by using minimally invasive microneedles on bioelectronics with thin-housing nanoparticles. In this case, we integrated the microneedles and flexible bioelectronics on the surgical forceps. If you see the scale bar, the size, I mean the thickness of the surgical forceps is very thin. So in the previous case, to implant the device, we have to open skirt over large area, but in this case, instead of that large area brain surgery, we can make a very small hole on the skirt and insert this surgical tool through that hole to reach the target region of the brain. We used two types of microneedles. The first one is bioresorbable microneedle, and the second one is the light guiding microneedle. In the case of bioresorbable microneedle, it contains thin-housing nanoparticles. So it has a iron oxide core for MR imaging, and on silicon shell, we have PDT dye, photodynamic therapy dye, and conjugated chemo drug. And if we can deliver high energy photons like UV light, then PDT dye will generate RS, reactive oxygen species, to oxidize the tumor cell. Also, it can release the chemo drug for chemotherapy. And to achieve this one, this strategy, first, we make a contact of this biodegradable microneedle to the target region. Then the needle will be dissolved as shown in this image, and the loaded nanoparticles will be delivered to the target region. And after that, we can just switch the microneedle into the light guiding microneedle integrated on the LED. Then the LED will generate high energy photons like UV photons, and those UV photons will be guided by this microneedle. And this microneedle includes aluminum oxide microparticles. So with just microneedle, the light will be delivered to this vertical direction only, but using this embedded aluminum oxide microparticles, the photons will be scattered and spread over larger area. So we can properly deliver UV photons to these thin-housing nanoparticles. And also, the LED will generate heat, which will be beneficial for accelerated diffusion of the nanoparticles and also chemo drug. And of course, there is a temperature sensor which can monitor the thermal actuation process to prevent overheating. And these images show the application protocol. We can first apply the biodegradable microneedle to deliver thin-housing nanoparticles, and then we apply high energy photons and heating to activate the thin-housing nanoparticles and diffuse chemo drugs. And as a result, according to the in vivo animal experiment, the tumor volume could be minimized and survival rate could be extended. Although the survival rate the efficacy is a little bit lower than the implantable device, but still this is beneficial because it has an advantage of minimally invasive approach. So that can be used as a successful second follow-up treatment like a booster shot, and I believe that we expect that this approach can improve the therapeutic efficacy of the implantable device. Let me move on to the last topic for the fatal seizure part. Before going into details, let me briefly explain the concept of seizure and the status epilepticus. Seizure is a symptom caused by abnormal brain activity. And when seizure occurs, patients typically show the uncontrolled shaking movement or lose consciousness. And in many cases, such seizures just go away naturally within 2 or 3 minutes. But in some cases, seizures can last more than 5 minutes, which correspond to medical emergency called as a status epilepticus. So again, the status epilepticus is a single seizure lasting more than 5 minutes, and it is a life-threatening medical emergency, in particular if an immediate treatment, pharmaceutical treatment to suppress the seizure is delayed. So between 10% and 30% people who have the status epilepticus can die within 30 days due to the brain damage. And it makes up about 1% of entire people who visit the emergency department. So the fatality is quite high, almost 30% people can die. And the number of the patient is quite much. So it is important to develop a new device for the treatment of seizure, as well as the status epilepticus. And again, previously, we worked on the development of the high-density electrode array, which could measure the intracranial EEG signal from the brain, epileptic brain, and it could map a spider signal when seizure occurs. And it is very beneficial to figure out the seizure focus. And that seizure focus can be precisely removed by the surgery. But even after treating many epilepsy cases, still intractable seizures remain. Almost 30% of entire seizure corresponds to intractable seizures, multifocal seizures, something like that. And such intractable seizures are resulting in status epilepticus. So we need also to work on the development of a new device for a status epilepticus feature continuous monitoring of EEG and fast delivery of the appropriate anti-seizure drugs. So if we have such a device, then we can monitor the patient EEG continuously. And if seizure is detected and lasts more than 5 minutes, diagnosed as a SE, then immediate anti-seizure drug delivery can suppress seizure and SE, then the fatal outcome can be prevented. However, currently that kind of device is not available, so we fabricated this kind of system, soft implantable drug delivery device, which is wirelessly integrated with the wearable EEG monitoring device. This schematic illustration explains how the system works. We monitor patient EEG continuously, and if seizure is detected and lasts more than 5 minutes, then emergency alarm signal, command signal is sent to this watch type power transmitter. And underneath this antenna, watch type power transmitter, a soft implantable, miniaturized implantable device is located. And this watch type power transmitter will generate strong radio frequency signal, which will deliver RF power to this implantable device. Then immediate drug release will be made and will suppress seizure and SE. This shows the EEG sensor mounted on the forehead and the watch type power transmitter. And there is an antenna, and underneath this antenna, this device is implanted subcutaneously. And this device has small size and soft mechanical characteristics because we move the sensing function to the wearable EEG sensor, and also power supply function and the control electronics to this wearable watch type power transmitter. So the implantable device needs only drug reservoir, which contains drugs, and ultra thin antenna and a little bit of the LEDs. So LEDs were integrated to indicate the drug delivery status, and the antenna receives the RF signal to transfer power wirelessly. And thus, the entire device is soft, so it can be deformed easily, and the size is small, so with minimum incision, we can implant this device under the skin. This slide shows the wearable devices. We integrate chips on the flexible PCB for wearable EEG sensor, and this device communicates with the wearable power transmitter through Bluetooth. And this wearable power transmitter has an antenna and sends power wirelessly using RF communication. And when RF signal is delivered to this implanted device, then water dissociation occurs inside the drug chamber. And when water inside the drug chamber is dissociated, then gas bubbling will occur. And due to this gas bubbling, the internal pressure of the chamber will be increased, and the outlet will be ruptured, and the loaded drug will be released within 3 minutes. So entire loaded drug will be released within 3 minutes, and so fast, rapid drug injection into the bloodstream can be made. And we tested the safety of the device systematically. First of all, we confirmed the SAR, specific absorption rate, which should be lower than federal regulation. And actually, our device SAR was lower than Apple iPhone 8. And also, we tested mechanical safety, robustness of the device through severe bending, systematic bending and pressing experiment. And even after repetitive bending and pressing experiment, the device showed no mechanical fracture or malfunction. And we applied this, we demonstrated this system using animal model. We induced status epilepticus in mouse model and measure EEG using our device. And when SE was detected, the command signal was delivered to the power transmitter, and this power transmitter generated radio frequency signal to transfer power to this implanted device, and immediate drug release was made in the experimental group, while negative control group, this system was not applied, so the immediate drug delivery could not be made. And according to the result, all mice in experimental group survived after 72 hours, but in control group, all mice were dead within 24 hours due to the brain damage. And we confirmed the brain damage using advanced blue. So we injected the advanced blue through intravenous routes. And in the case of control group, due to the brain damage, including the damage in blood-brain barrier, advanced blue was delivered to the brain tissue and dyed the brain tissue as blue. But in the case of experimental group, since we suppressed the SE and prevented the brain damage, so BBB was sturdy and protected the brain, and advanced blue could not be delivered to the brain tissue. And this movie shows the overall scenario of the application of the device. So high-risk patient can implant the device and wear devices to monitor EEG, and if seizure is detected and lasts more than 5 minutes, then the alarm signal can be sent to this power transmitter, and RF signal will be generated, and power will be delivered to the implanted device to release drug immediately, and then seizure will be suppressed. And we demonstrate this one using animal model. This shows the user interface made by Rapid program and shown on the mobile device of the user. When seizure is detected and lasts more than 5 minutes, the alarm signal alarm message will be sent to the nearby caregiver, and the RF signal will be generated, and the immediate drug delivery will be made. So the seizure and SE can be suppressed, and the mouse can survive from the SE. Lastly, considering potential human application, we try to scale up the device. So actually, we have two options. We can use small size device, as shown in the previous slide, with the highly concentrated drug, or we can use just conventional low concentration drug with increased volume of the drug reservoir. In this case, we fabricated a larger size drug delivery device, and we used a human SEEG signal collected from the patient, and this signal was detected by the system, and alarm signal, alarm command signal was transferred to this power transmitter, and power transmitter generated RF signal to release drug in the ex vivo experiment. Finally, we have been working on nanomaterials-based software electronics, and today I focused on devices for heart disease and brain disease, and we hope to continue our research effort to solve unmet clinical challenges using our technology. That's it, and thank you for your attention. Thank you.
Wonderful. That was an amazing presentation, Dae-Hyeong. Just an incredible spectrum of great ideas, interesting topics in material science, with mechanics playing an important role in many of those systems. So we very much appreciate your presentation and your willingness to participate in this EML webinar series. So I think we have a large number of panelists with us, and we can open up for questions and answers. But maybe I'll begin with a couple of my own. So these concepts in transient electronics appear to be very powerful, and your application in this context of treating tumors in the brain seems very exciting. I mean, when you talk to clinicians, how do they think about risks associated with the processes of bioresorption? And I think it's kind of a new concept here in the US. The FDA doesn't really know what to do with it because, you know, this kind of technology hasn't existed before. There are passive biodegradable drug release vehicles, as you know. People have explored biodegradable intravascular stents, but the idea of electronics, you know, that are biodegradable is totally new. And so what do you hear from, you know, your collaborators in terms of risk profiles? Do you have to worry about fragments, you know, toward the tail end of the degradation process? What are the thoughts? Do they feel like this is going to be something that can translate to humans? And you know, if so, what are the key risks? What are the challenges?
That is very wonderful question. First of all, the speed of the bioresorption seems to be important. If materials such as magnesium can be dissolved rapidly, then the local concentration of magnesium can be higher than the allowable limit. So that may be toxic. But since we are using proper encapsulation, so rather than just exposing magnesium or silicon to biofluids, we always use a proper encapsulation such as PLA or PLGA, which shows much slower bioresorption speed. So it seems to be fine. According to our experiment, it took almost 4 weeks to, more than 4 weeks actually, to dissolve the implanted magnesium devices. So it seems to be fine. And clinicians say that softness is very important actually. Since the toxicity is also an important issue because local immune responses can be caused by the local toxicity. But more than that, if the device is stiff, due to the micromotion of the brain tissue, seizure can happen. And after brain surgery, that seizure may not stay as a once, rather it can become repetitive epileptic seizure. So softness of the device, which can minimize the mechanical mismatch between the device and the brain tissue, is very important. And more importantly, we have to consider the benefit and risk both. And if the benefit is much larger than the risk, then it is worth it to try. And as you may know, the survival rate, 5-year survival rate of glioblastoma is 10%, although we are doing a lot of efforts from the surgical resection to radiotherapy to the chemotherapy. So 90% of people are dying after 5 years. So if the risk is not much, it is worth to try. That is the opinion of the clinicians.
That's a very, very good point. I'm glad you highlighted that. I'm also glad that you highlighted the importance of mechanics, soft mechanics in these platforms. As you know, for future publications, definitely think of EML as a target journal for papers, you know, on the mechanics. If it involves mechanics, I think it aligns well with our journal, and we would love to see submissions from your group. So it looks like we have a large number of folks with their hands raised. So I'm just going to go one by one. Maybe start with Zhigang. Do you have a question for Dae-Hyeong?
Yeah, thank you, John, for promoting EML. Yeah, wonderful. I have a question about your demonstration of glioblastoma treatment. Now, I see in your slide, I understand it's a demo of a concept using this platform, but I just want to know specific how effective it is. Now, this TMZ. TMZ are the standard treatment today is you have a drug, you take drug by mouth every day for 6 months. Now you propose to put it on the you directly on the tumor, right? So
yes.
Right. Now, what difference does it make in your experiment? You haven't done it with a human, right? Just
That's true. Yeah, we only tested with mouse model and canine model. And temozolomide is the most famous chemo drug for glioblastoma, and it has a higher delivery efficiency than other types of drugs.
Right.
However, still, due to the BBB, the delivery efficiency, due to the blood-brain barrier, the delivery efficiency is limited. Typically, tumor has leaky blood-brain barrier because of the tumor cell growth. The BBB is more leaky than other regions of the brain. Even considering that leaky BBB, the delivery efficiency is not very effective. That's because the gliadar wafer showed very good efficacy. Actually, the intracranial drug delivery can last less than a week. But even with that 1 week intracranial drug delivery, 6% survival rate could be improved to 20% survival rate. So that proves the efficacy and the efficiency of the direct intracranial drug delivery. And what we wanted to do is to include our biodegradable electronics to manipulate the intracranial drug delivery more efficiently. So we use some chemistry to make a conjugation between the drug and the biodegradable polymer, which can us to use which enables the sustained long-term drug delivery. And also we used a magnesium-based heater with wireless actuation for controlled drug delivery, as well as the deeper penetration. Deeper penetration is also a very important issue. Without such thermal activation, the penetration depth is very shallow, although glioblastoma is very infiltrative into the normal brain tissue. So in the field of GBM treatment, always deep penetration is a hot topic. And that's the reason why we can utilize wireless electronics to enhance the penetration depth. Although it is not perfect with current result, we are trying to improve the penetration depth further and further up to centimeter scale penetration depth.
Yeah, there is also related clinical observation for several decades about this. I don't know if you study this. So when patients come in, they do a test to look for this marker, methylated or unmethylated. So the classical delivery by mouth is known to be not effective for unmethylated patient. Have you tested this aspect? Would delivery method really resolve this issue, or is it really just chemical, methylated or unmethylated?
Frankly speaking, I'm not familiar with that strategy, but as far as I know, there are many different types of approaches to increase the systemic drug delivery through the BBB. So first challenge is to pass through the BBB, to pass through the blood-brain barrier. The second approach is to achieve higher targeted drug delivery using some target moieties. But unfortunately, glioblastoma shows very high heterogeneity, and they are scattered a lot. So depending on tumor type, some tumors are concentrated at a small region, but unfortunately, GBM is highly infiltrated and scattered. So targeted delivery is very, very challenging, in addition to the heterogeneity due to the scattered shape of the GBM. The targeted delivery is very challenging. So that's the reason why many chemists are working on the new on the development of a new method for efficient drug delivery.
Thank you.
Great, thank you, Dae-Hyeong. It looks like we have like 8 or 9 people waiting to ask questions, so please have patience. I'm just going to go one by one. So, so Philip, you're up next. Would you like to ask your question?
Yeah, great talk. I really enjoyed it. My question is related towards the electroceuticals, right? The last part of your talk. So for devices that elute drug inside the body, often there's a couple of, especially for chronic applications, there's 2 problems, right? One is the drugs often don't last at body temperature. Maybe you can comment on this with your application. And then second, the electronics also have limited lifetime in the body. That's a kind of the other kind of question regarding this. But more broadly, also with the availability of drug-eluting devices inside the body, do you think that drug manufacturers or drug researchers can refocus their efforts on different types of drugs that make specific use of this route of delivery? And also, what do you think about the the patient has to wear a lot of gadgets. What's the patient acceptance of these type of devices? So lots of questions in one. Sorry about that.
Thank you for the excellent questions, although it is very long. I'm not sure if I can remember all the questions. First of all, yeah, drug seems to be fine. Yeah, although we didn't test over a year or something like that. We just tested a month inside a animal mouse. So that may not be enough. So we need further study. But at least a month, the drug seems to be fine. And electronics, since it is just an antenna, it seems to be fine. But the more serious issue is actually was in biofouling. The electronics was fine, but due to the gas bubbling, internal pressure will be increased, and that increased pressure should break the outlet of the drug chamber. And we wanted to make sure the robustness of the outlet. So we didn't want to accidental, unwanted breakage of the outlet due to external pressure or motion of the patient. But in addition, we found out that after a month, severe bowel fouling can occur on site on top of the drug outlet. Luckily, when we tested our device, although there is a biofouling layer, if the rupture is properly made, then the drug diffused through the biofouling layer and could be delivered to the bloodstream within several minutes. And also there was a report from the Langer group. They tested implantable drug delivery for 6 months in the human body, and even after severe biofouling, they also could deliver a drug properly. Yeah, so that and the next question was drug. Yeah, I think that implantable drug delivery device has a disadvantage in terms of the surgery. We need, although the surgery is not very large, we can just make a small incision, less than 1 centimeter, and insert the device under the skin. So actually clinicians, surgeons don't think that this is a serious issue. However, in comparison with wearable devices, for example, transdermal drug delivery device, still it is a burden to the patient. But the advantage of the implantable drug delivery system is that we can use a variety of drug types. To pass through the skin barrier, there is a clear limitation in choosing drug types. And the drug delivery speed and drug delivery efficiency are much lower than the implantable device, although we need immediate treatment in the case of emergency status epilepticus case. So that flexibility can enables us to use a variety of different types of drugs, which can be beneficial to the patient, and maybe that motivate drug companies to develop a new type of device for specific disease models. And is that all? I forgot questions.
Yeah, that answered almost all the questions. I think the patient acceptance also relates somewhat to having to wear a headband, having to wear a type of transmitter that needs to be also topped up with charge all the time, right? Otherwise, because you have to have the device available at any given time. So I don't know if you looked into this. I mean, it's obviously at the animal stage model, but I think those are probably barriers that are hard to overcome for these kinds of devices.
Yeah, that's true. That's true. It is very important, and it requires a lot of engineering, I guess. So I think that the technology is already available. If we design special chip which can all the electronics function into a small chip, then maybe we can miniaturize the entire size of the device a lot. In our case, we purchased the chips separately on the market, and my students designed the flexible PCB and assembled all these chips on the flexible PCB. So the device size is very large. But if we can use that specific chip, specially designed chip, then probably we will be able to minimize the device size. And also, battery is also an issue. The battery size is large. So we need to try minimize the power consumption. And luckily, already such technologies to minimize the power consumption are available, although we cannot use that right now in the academic side. But if we think about companies, then and if they intend to develop a miniaturized, user-friendly device, then probably we will be able to achieve a much smaller user-friendly device, I think.
Excellent. Thank you.
Thank you.
Yeah, that's a really good point, Dae-Hyeong. A lot of the technologies that you're developing are well aligned with the consumer electronics gadgetry industry, and there's a huge driving force around miniaturization and improvements in power efficiency and so on. So I think the patient burden associated with these kinds of devices will decrease over time through those efforts. So it's a good point. Jimmy, you have a question.
Thank you, John. Dae-Hyeong, beautiful talk. A lot of cool ideas, cool devices. I have 2 points. One is more of a comment. The other one is a question. The comment I have is related to the first topic you mentioned. You have this conducting rubber wrapped around the heart in order to stimulate the heart contraction. Indeed, heart muscles contract in a synchronized way, but they don't contract simultaneously. We have Tahir Saif over here. He's an expert of this. And we also published a paper in Science Advances in collaboration with Zahi Cohen-Carney at Carnegie Mellon when I was there. And we showed that if you have a heart or a cardiac muscle of roughly about 100 micron diameter, the signal delay from one side of the muscle to the other side is roughly about 10 milliseconds. So it's not simultaneously. Whereas conductors, the speed of transmission is much, much faster. So that's a comment I think you'll probably need to pay attention to. The question I have is also related to the heart repair, your contact printing of cell sheets. And heart is essentially a mechanical device. So the contraction direction of the cell that you printed onto the heart to repair it is important. The direction of the contraction is important. Did you control the alignment or maybe when you print it, they all aligned along certain directions? If you do, how do you do that?
Yeah, thank you for the comment, first of all. And if I make some comments about the first comment, then yeah, that's true. So the cardiac signal spread through the Purkinje fiber network and from bottom to the top in the case of ventricle. So there should be a small time difference for the contraction of the cardiomyocyte at the bottom and the top. That is true. So previously, since we used mouse model, the heart was so small, so we could not change the stimulation sequence with some appropriate time interval. But in these days, we are working with either rabbit or pig. So, and especially in the case of pig, since the heart size is quite large, we are using multi-channel electrodes. So, and we designed the electrical stimulation protocol. It can be either local stimulation, biventricular stimulation, or multifocal stimulation with appropriate time delay and intensity manipulation. And we are not sure which one is the optimum stimulation protocol yet, because depending on the heart failure model, the outcome is a little bit different. But it is true that in comparison with the negative control group, if we apply appropriate stimulation, of course, which should be optimized for a specific case, then the pumping capability can be improved. So yeah, that's what we know until now. And that's the reason why we are continuing the research with large animal models. And the second question, the cell sheet therapy. So at first we tried to use cardiac muscle, and if you take a look at the cardiac muscle, then it consists of multiple layers of cardiac muscles, cells, and each layer has a perpendicular alignment. So we wanted to mimic that perpendicular stacked layer at first. But unfortunately, if we transfer print cardiomyocytes, that may cause some risk of the arrhythmia because of the abnormal myocardium contraction and the signal propagation. So instead of using that, in these days, people are using stem cells, induced iPS or the MSC. So in such cases, I'm not sure if the alignment is very important. But anyway, we can do alignment. We are patterning the graphene layer as a line and space. And on PDMS, the cell adhesion is very bad. However, on graphene, the cell adhesion is very good. So cell aligns following the line and space pattern of the graphene. So we can easily align the cell sheet. But yeah, that part needs to further studies for optimum cell sheet alignment and optimum cell type for cardiac regeneration.
Thank you. Thank you very much.
Thank you.
But just as a follow-up, Dae-Hyeong, I got the impression that these are more or less monolayers of cells, and I don't know if that's kind of ideal, or do you want to grow multi-layers, and what is the optimum thickness? And as you change the thickness, if you go to multi-layers, do you have issues in cell-cell adhesion and printing? And what are the considerations around those sorts of topics?
Mostly it is a single layer. We couldn't culture multiple layers yet, but as far as I know, in industry, when engineers culture skin tissue in large scale, they are culturing multiple layers of tissue. So the technology is already available, although we cannot do that. Since we only cultured single layer cell sheets, our transfer printing process was optimized for the single layer transfer printing. But I believe that although the thickness of cell sheet is thicker than current one, the transfer printing should show high yield, because that just needs crack generation during the preparation step. And regardless of the cell sheet thickness, crack generation may be made properly, I guess. Yeah, but we didn't try.
I was just wondering if you would get cohesive failure, the crack would run through the middle of the multi-layer, cell stack or something.
Yeah.
Try to bring mechanics into the conversation a little bit here. So.
That's true. That's true. Yeah, we need consulting from mechanics.
Yeah. If I remember right, Nanshu did some of the modeling on those studies, right? The transfer printing aspects of manipulating these cell sheets. Is that right?
Yes.
That's true. That's true.
Yes. Okay. Yeah, I remember that paper. It's very interesting.
Yeah. In fact, Nanshu contributed a lot for all the mechanics analysis.
Yeah. Just to clarify, you culture a single cell sheet or a single layer of cells, but you print multiple layers. Did you?
Yes. By repeat the transfer printing process.
Yeah. Okay.
Yeah.
So we typically use the 5 layers. Yeah.
Mm-hmm. Yeah, it's really fascinating stuff. Maybe we'll go to the next question. Hi, Chao. Han.
Yeah, this is Hai Chao Han from University of Texas, San Antonio. So I do cardiovascular biomechanics. So my question is related to the heart mesh there. So I think it's a very interesting idea to have this multiple spot, multiple stimuli to help synchronize the heart. I like that idea. So my question is about the cell sheet or the nano wire you put it there. So I think you can adjust it, right? So help the contraction. But then when the heart tries to dilate, so during the diastolic phase, I'm just concerned whether that mesh or the sheet will increase the resistance for dilation. So what does the clinical person or the cardiologist think about that? And also, have you measured how much resistance you're applying, you're adding to the heart when you add in the mesh there?
Yeah, that is a very important point. In our case, we just mimicked the modulus of the myocardium. So systematic modulus of our device to be matched with the modulus of myocardium. That's what we used. But more precise manipulation of the stiffness, elasticity of the cardiac mass is required. And according to recent report from other group in 2019 in Nature Biomedical Engineering, by carefully designing the viscoelastic property of the cardiac mesh, it can help contraction of the heart, as well as it can prevent dilation and shape remodeling of the heart from MI to heart failure. So yeah, that was purely proved by mechanics and animal experiments without any electrical stimulation function. So by properly designing the shape of the cardiac mesh as well as the intrinsic modulus of the material, it seems that it is beneficial in preventing severe heart failure progression. But the dilation is also troublesome. So we do not want to apply too much pressure because it will prevent dilation, diastolic stage of the heart. And in that case, patient will feel difficulty in breathing. And also it can cause arrhythmia because of the too much contraction of the natural motion of the heart. And actually, depending on cases, helping dilation is more useful than helping contraction. In such cases, spring can be inserted inside the chamber, and spring helps dilation rather than contraction. Maybe it can prevent contraction, but it can help dilation of the heart. So it depends on case and the patient condition. We can use either cardiac mesh that helps contraction or spring and located in the endocardium to help dilation.
Yeah, that's a very good point. So I know there are more than half the heart failure patients, they have diastolic problem. So their heart cannot relax, dilate enough during the filling phase. So we can adjust it to help the filling, the dilation, and think that will be useful for half the heart failure patient. Thank you.
Yeah, but there are pros and cons. If we put in springs inside the chamber, that can cause some clot formation and some side effects, micro damages, something like that. But yeah, as you mentioned, many people are suffering from difficulty in making dilation. So definitely, mechanical point of view, it is very beneficial. Yes.
So sorry, what's the clinicians, they think about you add a mesh, something there outside the heart. They feel it's safe or they feel scary, or what the kind of a feeling they told you?
So actually, mounting device on the epicardium seems to be quite safe, although it requires open chest surgery. So clinicians prefer minimally invasive surgery, actually. So if we can deploy spring using a catheter, for example, that is preferred. If we want to wrap the device around the heart, that it definitely requires quite large incision. And actually, there was a company that tried to approval of the cardiac rubber-based cardiac mesh from FDA, and they failed eventually. And the reason is because although there is benefits, the cost is too high because it requires open chest surgery. But the clinician point of view, there are patients, end stage of the heart failure patients. Their expected life is about 6 months, and they are waiting for heart donation. They need heart transplantation. And always the list of the waiting patients is very long. So before they get a chance to be donated, transplanted, they usually die. So in such cases, although the cost is very expensive, if it can last the patient alive more than a year or a year and a half, then the chance to get donation of the heart will be increased a lot. So in that point, clinicians, at least my collaborators, thinks that this device has some meaning to proceed the research.
Thank you.
Great. Thanks, Dae-Hyeong, for that answer. Thanks for the question. Xing, you're up next.
Yeah, thank you very much for the very inspiring talk, Dae-Hyeong. I have a follow-up question on the cell patch. When you transfer those cells on the heart, how are you going to make sure the cells adhere to the heart chamber? And the cells may beat synchronously in the patch dish, but when you transfer them on the heart, they may behave very differently. How do you make sure they do whatever the job they are supposed to do? Another thing is for the heart wall, it actually contains multiple cell membranes. One is this pericardial membrane, and in between there is a liquid layer. So do those cells have to penetrate those layers to access those myocardial chambers, or you have to do the surgery to peel off those membranes to access those muscles? Thank you.
Thank you for the question. And frankly speaking, we didn't try cardiac therapy in vivo. What we tested is muscle regeneration in limb scar model of the mouse model. And we just followed and adapted the result from Japanese groups. And they already tested the stem cell sheet therapy in the clinical trial, phase 1 clinical trial, and they confirmed the efficacy. That's the reason why they made a startup company and proceed commercialization. And at least the stem cell sheet transplantation shows efficacy, but again, that requires open chest surgery. Cell sheets cannot be transferred minimally invasively. We need to remove pericardium and directly transfer to the scarred region. So the cost is a little bit high. Although there is a benefit. So always we have to consider both benefit or cost. Yeah.
Thank you.
Great, thanks, Chang. Tahir, you're up.
Thank you, John. Very, very nice talk. Lots of very good discussions. So I have a couple of thoughts regarding the transient electronics, bioimplantable electronics. I think this is a hugely potential area. And given the access from the outside, either by RF signaling and optics, I'm wondering what are the actual limits of these bioelectronics or these transient electronics? So is it possible to have not only just the drugs that you have been discussing, but also add cells to them and have some feedback control that something can be read out from the outside? So to be more specific, I'm thinking whether we could have in the transient electronics, the iPS-derived cardiomyocytes or cardiac cells that can be used as a patch, and the electronics that go with it may train the iPS-derived cardiomyocytes to match their dynamics with the existing heart cells. And eventually, through this crosstalk facilitated by the electronics will dissolve, but the new patch would become part of the cardiac cells or the heart. Is this a possibility? In other words, can this transient electronics not only be loaded with drugs, but also living entities that could be eventually part of the body? So in case of glioblastoma, is it possible to have the same chips, but now loaded with maybe stem cells, which might replace some of the damaged cells, or maybe even proactively do something onto the glioblastoma? So the stem cells would have to be trained. So in other words, these transient electronics can they be also biohybrid? Can they be also living entities?
Yeah, that is actually a quite good idea. And what we tested is to transfer print cell sheet on top of the implantable electrode.
Mm-hmm.
So for neural interfacing, if we just make a direct contact of the device, electrode, to the target tissue, then always inflammation occurs, and due to the immune responses and macrophage collection occurs. That can be due to the either partially due to toxicity or partially due to mechanical friction force or stress. But when we transfer print 5 layers of a cell sheet on top of the device, then still we could record electrophysiological signal, and also we could transfer the electrical stimulation signal to the target tissue. But due to the interfacing cell sheet layers, the immune response could be significantly reduced. So much lower macrophage collection and immune responses could be observed. So that's what we know. That's what we already published. But your question is to integrate stem cells, for example, with the device and implant that on the heart surface or brain surface, things like that. And as far as I know, I'm not an expert too much, but as far as I know, rather than making these transplanted cells to be vascularized with the existing cells, paracrine effect. So when cell dies, they emit some valuable proteins and things like that, and that is very helpful for regeneration of the cardiac muscle and maybe brain. And it's quite difficult to externally control the microstructure of the neural network or cardiac muscle. If that precise control cannot be made, then side effects can occur. Typically, seizure in case of brain, and arrhythmia in the case of heart. So I think that mostly paracrine effect is being used for the regeneration therapy. Yeah, and but yeah, I think that is very interesting. We can attach cell sheet on the device and apply some stimulation to accelerate the vascularization and the cell regeneration, and that definitely will be helpful for muscle regeneration, since muscle is much simpler and has aligned the direction. Then electrical stimulation will be definitely helpful for accelerated muscle regeneration.
Thank you.
Yeah, very interesting questions and suggestions, Tahir. I strongly suspect that some of those directions are being pursued currently in research labs around. So very, very insightful. Yi Hui, hello. You're up next.
Hi, Dae-Hyeong. Wonderful talk, very inspiring and informative as always. I have 2 questions. Since this is an EML webinar, I'm gonna ask a mechanics question first. So we know that when the biointegrated electronic device is used inside the body for a long time, the fatigue and the degradation of the device system due to the coupled humidity, chemical reactions, and mechanical loadings could be a potential issue. And so in your case, if we think of the your cardiac mesh device integrated with the heart continuously for a week, for example, for health monitoring, then the device system needs to undergo 0.6 million cycles of dilation and shrinkage due to the heartbeat. And also the environment of the body fluids can also potentially affect the device performance through the chemical reactions. So I'm wondering if you have studied or evaluated such long-term stability and reliability of your cardiac device. And so this is the first question. And the other question is related to merging artificial intelligence with biointegrated electronics. We know that the field of AI has made tremendous progress in recent years. So what new opportunities would these advances in AI bring to the field of biointegrated electronics? Can you provide your perspectives on this point?
Yeah, thank you for the nice questions. In the case of long-term reliability, yeah, we could not confirm that yet. We tried implantation for a month, but at least a year in vivo implantation should be proceeded for potential clinical translation. And even after 1 month's implantation, biofouling and scar cell collection occurs. Think about the surgery. Always after surgery, some scar cell collection and fibrosis occurs. So likewise, we could see similar thing. But the good thing is that the device can still apply mechanical force as well as electrical stimulation. But we need to solve such a long-term biocompatibility and make sure that the device is properly working over extended period of time. That's what we need to do in the future. And AI, yes, we couldn't work on that much, but yeah, definitely AI should be very useful tool to train the device to be more smarter. Yeah, many people, it seems that many groups are using AI algorithm to collect biosensing data and analyze the biosensing data. So optimize the feedback stimulation, things like that. So yeah, in the future, definitely we should work on that, and that should upgrade the entire protocol of the biointegrated electronics. I think so.
Okay, thank you. What about the mechanical failure and the fatigue of your stretchable silver nanomaterials? Have you studied the performance under cyclic loadings of, for your silver nanomaterials?
Yeah, that is another good question. It is related with the movement of nanomaterials inside the viscoelastic media. And in the case of SCBS, it is true that some nanowires are entangled each other and segregated each other under long-term mechanical repetitive cyclic test. So we are trying to use SCBS, TPU, or other types of polymers, which has better stability and a little bit has a little bit higher modulus. But by combining some special design, that issue can be solved. And by optimizing the polymer and also optimizing ligand of the nanowire. Okay. Ligand usually is located on the surface of nanowire, and by controlling the length of the ligand, the micromotion inside the viscoelastic media can be totally different. So by optimizing the ligand property as well as the elastomer type and property, we believe that we will be able to solve that issue in the future.
Great. Thanks for your answers.
Thank you.
Yeah, just to follow up on that, Dae-Hyeong, I think a lot of the community in soft electronics, bioelectronics, one of the goals around the mechanical properties are low modulus, high elongation, elastic response to large strain deformation, that type of thing. But what about other kinds of mechanical properties in the materials or the composites, like viscoplastic behavior or superplasticity, hyperelasticity, maybe materials with defined viscoelasticity for damping sort of mechanical motions, interfaces with are there things beyond just low modulus elastic properties that would be interesting for the mechanics community to think about, for different use cases? Like, for example, I think the Stanford group had a platform that was viscoplastic, so it was able to sort of follow patterns of growth. And there you don't want, an elastic restoring force to maybe constrain the growth of tissues, but you want the device to follow those dimensional changes. I mean, what do you see in the sorts of projects that you're working on that would go beyond some kind of more elaborated set of mechanical properties?
Yeah, that is very important point. I think such as sapling polymers or hydrogels have been researched for that purpose. In such cases, a dynamic rebonding of hydrogen bonding or other types of bondings can adapt the shape of the device locally and also minimize the mechanical fatigue or stress applied to the target interfacing tissue. Especially device implantation around the spinal cord or peripheral nerve, where a lot of motion or deformation is involved. Such viscoelastic property is very beneficial. And also, as I mentioned, in the case of cardiac mesh, one group has used viscoelastic hydrogel, injectable hydrogel as a patch, just simply mechanical wrap around the myocardial infarction heart, and they could prevent dilation and heart remodeling and prevent the progression of the heart failure. So and definitely in the case of brain, always micro motions are involved, and low modulus is also important. So in such cases, hydrogels are a very promising material. And such as supramolecular materials can be actively used in various applications, from brain to peripheral nerves to the heart. And we are also studying that part. We still, until now, we still stick with the conventional elastomers such as PDMS or SEBS, but at the same time, we are studying hydrogels or mixture of the elastomer and hydrogels. For example, hydrophilic polyurethane can be mixed with hydrogel to show the viscoelastic property and softness of the hydrogel, as well as the mechanical toughness of the elastomer. So we can find the ideal property in between. So such efforts should be needed in the future.
Yeah, fantastic. As you know, we have a couple of hydrogel experts on the panel right now, so I'm sure
that's true.
Hear those comments and thoughts. Yeah. All right, Nanshu.
Yes, I also want to help Dae-Hyeong answer John's question, like what we normally do. So John has a very good question, like what kind of ideal mechanical properties we desire for future soft bioelectronics. And John mentioned like this plasticity with growth, and John has his own work with Yi-Hui on the hyperelastic skin, like stiffening epidermis layers. Those are also good examples. And ultimately, I think fatigue, as Yi-Hui mentioned, has to be studied, although we try to avoid the very dirty work as much as we can, but it is very important. Zhigang can speak to that. And ultimately, we care about damage under high cycles, and we are also needing damage-resistant material or ultimately self-healing material that can self-repair even if damage occurs. Jian Liang and many others are working in this field. I also want to add that the desirable mechanical properties highly depend on the interface we are trying to maintain. So the reason we care about the softness and the thickness is because we are trying to conform to highly curvilinear and dynamic biosurfaces. On the surface, we have coupled mechanical, electrical, optical, thermal, biochemical interactions, and they one affect the other. So because of that, we have to study the multi-physics on such interface. And in addition to like conformability and long-term attachment, we can also leverage those kind of weak interface. For example, the cell sheet transfer. So in that case, we want to first be able to create initial cracks between cell sheet and graphene through this one-step manipulation. And then because we have pre-existing crack, we can easily deliver transfer print cell sheet onto target substrate. So in those cases, we actually take advantage of interface failure. And of course, John has this speed controlled transfer printing. That's another very good example. But another example would be biofouling. So when we have repetitive mechanical motion, like the heartbeat or the brain motion, they actually induce fatigued interfacial detachment between the biofilms and our devices. So in that case, those mechanical fatigue helped release some of those biofouling effects. And ultimately, I think we mechanicians will have to deal with more like multi-physics, and material scientists and device scientists, they will have to appreciate mechanics to be successful, just like what everyone here has been trying to do. I hope I helped a little bit. So, and I have a question for Dae-Hyeong. Although I'm very familiar with your work, of course, I always get new inspiration after listening to each of your talk. So one question I have, because recently I'm also working on some porous conductive nanocomposite. I learned that if we use highly porous elastomer doped with conductive filler, we don't need to have a lot of doping concentration. Because of the big pores, they will concentrate the elastomer and the filler into like thin ligaments or thin layers. Because of that, we can significantly reduce the doping concentration, but still reach the percolation threshold. For example, we use nickel foam as a template, and we create conductive carbon nanotube doped EcoFlex. And the doping concentration for the percolation threshold is only about 0.3 weight percent. So if that is the case, is that useful for your epicardium mesh, conductive mesh? Because there you try to maintain softness while increasing the conductivity. And why you considered silver nanowire instead of like those carbon-based fillers, because we also know if we have a hybrid form factor of the filler, like a nanotube combined with graphene flakes, those kind of 1D, 2D morphology could also enhance the connectivity. That's my question.
Actually, that kind of porous nanocomposite should be very, very beneficial for the implantable biomedical devices, because, as you mentioned, we can minimize the loading amount of the conductive filler while maximizing the elasticity of the composite. So that should be very beneficial, and we actually are working on that part also to maximize the material property, mechanical material property, and minimize the loading amount. What was the second question?
So what's the difference between like carbon versus silver nanowire?
The reason why we used silver nanowires at first was all because of the extremely high intrinsic conductivity of the silver nanowire. But as I mentioned, the silver nanowires have many issues, particularly in oxidation and also toxicity. So that's the reason why we used cold shell type nanowires. And in these days, we are trying to use another metal, noble metal-based nanomaterials to solve that toxicity issue. And such low resistivity is also helpful to minimize the impedance and contact impedance. And I believe that a porous structure that you are working on should be very beneficial also to minimize the contact impedance. And actually, low impedance is important not just for the high signal to noise ratio recording, but also to minimize the electrical stimulation fatigue for long-term stimulation. Low impedance is very important. So,
we also read your recent porous hydrogel epidermal electronics paper. Very inspiring. Thank you.
Thank you.
Yeah, great work. Thank you for the question, Nanshu. So we'll go to Junsu. You're up.
Thank you. I'm Junsu, a graduate student from Harvard. Thank you for the talk. I clearly see how you go further from your PhD topics. As you mentioned in the informal interview, there are a lot of details in certain achievements. So thank you for showing that path. So I have two questions. One is about the outlets. For example, in the case of the heart valve, not heart, and the curvilinear serpent types sensing around the heart. So you said there should be multiple wires, there should be multiple signals and stimuli to give some local difference. Then I was thinking about the resolution, what resolution should be there to fully control the local motion, distributed motion. If it is quite dense, for example, if we need 100 by 100 matrix, then we need about 10,000 outlets, which will be very bulky at the end. Of course, we can solve this issue by using transistor at the matrix, like our display. Then we may need stretchable transistor. All these questions come from there. So what is the required resolution, ultimate goal in terms of the resolution, and what could be the solution about that? That's the first question. The second question is about the interface between heart and electrode. It seems there is no further adhesion layer between the elastomer and the heart, and because of the repeated motion, maybe they will slip a little bit, change their location, stimulate different location over time. So how do you deal with this kind of change over time? And what could be more mechanical challenging mechanical problems in these kind of devices? Thank you.
Thank you for the excellent questions. In the case of resolution, actually we didn't talk about much about the resolution of the cardiac mesh, although we discussed a lot about the resolution of the brain interfacing device. In the case of epicortical mapping device, usually 300 micron spacing between electrodes seems to be ideal. The smaller resolution may not be very beneficial because of the spread of the electrical signal through the conductive biofluids. But in the case of a heart, we do not know exactly, since we mostly tested our device in mice or rats or rabbits. Large scale, high resolution test should be proceeded in pig model. And we tested the pig model only several times, so it is definitely not enough to determine the resolution. Until now, we have, for the pig model, we have almost 50 channel, 80 something channel. And that was good enough for the demonstration and controlled electrical stimulation. But probably there should be some ideal point. We may need to increase the number of channels further. Or we can focus the location of channels to specific region of the heart, such as LV. Usually, the ischemic injury occurs in the left ventricle, so we can focus LV and increase the density of electrodes in LV, and maybe use a little bit sparse electrode spacing in LV region. And such a strategy can be used, but it is true that further studies are required. And the second question was how to fix the device on the hot surface. That is also challenging issue. We usually sutured the device on the surrounding tissue, and that was good enough for short-term demonstration and recording, but it slips. And since we want to measure local EKG, if the location of the electrodes changes, that is troublesome. So what you can use is bioglue. We can glue some specific region of the electrode to the hot surface, but if we use too much glue, then that can restrain the natural motion of the heart, and that can be a potential focus of the arrhythmia. So that may not be an ideal case. So recently, what we are using is adhesive hydrogel. Instead of the bioglue, if we use adhesive hydrogel, then due to the low modulus, mechanical deformation occurs while it fixes the device on the hot surface.
Maybe including our group, some groups who works on the adhesive hydrogel could help to fix the location of a lot of implantable devices. I have one more follow-up questions. So for hearts, maybe it's not the resolution, required resolution is is already good enough. I don't know, but for brains, obviously we need a lot of electrodes to get more information at the same time. So I easily expect a bunch of outlets. For example, if we need 10,000 outlets, signals at the same time, the outlet from our head will be quite thick. Is there any ultimate solution on it, or is it okay to have a quite thick outlet from our brain?
Yeah, in the case of short-term implantation in the clinical area, already that kind of approach has been used for 2 weeks implantation or 3 weeks implantation for seizure mapping before seizure surgery, brain surgery. But yeah, definitely, if we consider the daily lives, that is not ideal, and fully implantable system is required. And already there are several reports for fully implantable system that includes power supply unit and wireless data transmission. Yeah, I'm not sure how many channels can be included in that fully implantable system. But yeah, I think it's just a matter of engineering. We can work on chips and electronics to solve that issue if needed.
Thank you so much.
Yeah, I would agree with that last comment. I think Neuralink, Elon Musk's company, they have around 3,000 channels, these filamentary probes, and they use a Bluetooth wireless-based system. There's a lot of data aggregation that's happening locally, so they're not streaming the full, sampled waveforms at each measurement node. There's just counting spikes, so you're just counting timing events. So you can reduce the wireless data transfer bandwidth requirements very, very significantly. And we're part of a DARPA program led by Ken Shepard at Columbia. It's 56,000 channels. We'll see. I mean, it's DARPA hard type of work, but I would say it's a doable sort of engineering challenge. But I think these are good questions, good issues. So this is a great discussion. Unfortunately, I don't know if you can tell from my background, I'm in a hotel room and they're trying to kick me out right now. And so I have like 3 or 4 minutes left. So I think I'm going to have to sign off, unfortunately. Maybe Ting, Sulin, or Jimmy, you guys can pick it up from here. I don't want to disrupt the rest of the discussion. It's been a great dialogue. And Dae-Hyeong, thanks so much for joining and presenting your wonderful research results to this broader group. And we have a lot of really productive groups represented by the panelists as well. And I want to emphasize, most of these groups are sort of materials in their orientation, maybe biomedical engineering, but there are really important mechanics concepts associated with a lot of this work. And I would just encourage everyone to think about EML, as a journal destination for the mechanics related aspects of your research. I think, to your benefit, the editors respond very quickly. So we'll prioritize your manuscripts and make sure that things happen in a very expedited way. That's one thing to consider. The other is that I think by publishing in EML, you'll gain exposure to a slightly different community than you would if you published in the more typical places, advanced materials, nature communication, that sort of thing. There's a very vibrant and robust and large and active community of mechanicians. And I think they're highly interested in this sort of work, and you'll be able to get some exposure, for your research to that community. And I think it's an additional outlet, that I'd encourage you to keep in mind. So with that, I'm going to thank all the panelists. Thanks to Jimmy for setting this up, Zhigang for launching this webinar series to begin with. Thank you, Dae-Hyeong, again, and I apologize that I have to drop off, but they've been knocking at my door asking when I'm going to check out, and so if I don't do that within 2 minutes, I got to pay for another night stay.
Okay, all right, thank you so much, John.
Okay, all right, good to see everybody. Okay, bye-bye.
Bye.
Okay, Tang, you're next.
Okay, so thank you, Dae-Hyeong, for this wonderful talk. I'm quite sure your talk actually attract a tremendous interest, and we indeed in the another live streaming platform in WeChat, there has been more than 7,000 viewers of today's webinar. Those people, even though it's quite late, majority of the viewers, I assume, of there is in Asia. They don't have the opportunity to ask questions or raise hands. So I guess my next question is indeed on behalf of those audience in a different live streaming platform. I have been watching the live chat box in that platform. There were quite some active discussions while you were giving the presentation. At a certain point, some some new audience I was actually asking, is the speaker a medical doctor? I guess that's when you were presenting the research results related to the treatment of the disease or versus the, I mean the in vivo monitoring of the biomedical device in operation. So the question shaped during the chat box over there is somewhat along this line. When you, I'm quite sure you learned a lot when you were in John's group. When you started your own individual, independent research career, you established a lot of collaborations with people in the medical field, right? In the physicians, doctors, and that's actually is growing sustainably and also thriving, as you can see from your presentation today. People were actually interested to ask you that when you first started, how did you overcome the initial barrier to start the collaborations locally at, for example, near SNU and the local doctors in Korea? Because I'm quite sure John set up the role model how to approach those field and those collaborators in quite different field in his group. But when you started your own group, how did you overcome this initial barrier, and how do you keep this going and keep this sustainable? I think that's a question that quite some younger researchers are interested to know.
Yeah, I was very.
Let me a little bit more specific. Did you go after them, or did they come to you?
I think both. Yes, sometimes my senior advisor in my department, I mean another professor in my department, a mentor professor, introduced me a clinician who can collaborate with me, and again he introduced other clinicians to me to figure out some details in clinical area. Also, sometimes some clinicians approached us after reading our publication. They always brought very nice ideas. So usually using the same device, we could expand the application to different disease types or different specific cases of the same disease type. So actually both helped us to make very good collaborations with clinicians.
So did you ever encounter the situation that you need to try hard to convince the medical doctors or physicians that, okay, material science and can make a contribution to offer unconventional solutions to the, for example, the diagnosis or the treatment of the certain diseases?
In my case, I was lucky that all the collaborators in clinical area were very passionate and eager to apply our devices and materials to animal models. Maybe the other clinicians in the hospital are reluctant to apply our devices, but in my case, my collaborators persuaded other people to proceed our experiment. So yeah, I was very lucky in that point.
Thank you.
Thank you.
Thank you. Sulin?
Hi, I don't have a question, and it's a wonderful discussion. I just want to have a comment following actually Dae-Hyeong's discussion. So we know that for any bioplannable devices, biocompatibility is a concern. In your case, So when you print the cardiac tissue and put it on the cell, on the heart, the compatible dynamics is obvious a concern. How this printed tissue is stretched, which direction is stretched, whether they are synchronized with the real heart beating. So those are the concerns. I think Tahira brought up a very good question. Can we train the tissue before you put it on the heart, right? I think there are a lot of efforts already already there in academia. So we know that cells are very responsive. Cell can sense all kinds of cues from its surroundings and respond differently to these cues. So somehow you can impart all kinds of cues like topographical cues, electrical cues, and material cues, all these kind of things, and elicit or program the dynamics of the cells. And if we can do that, I think that will change completely maybe the landscape of the devices and the printed tissue. Yeah.
That is a wonderful idea.
Yeah.
So
yeah.
Hybrid system.
Yeah, hybrid system. Yeah.
Yeah. Okay, thank you very much. And I just want to remind everybody that it's 1 a.m. or past 1 a.m. in Korea. So Dae-Hyeong, it's wonderful to have you give the talk, and we all enjoyed it very much. And I think John said it very well. So let's give him a round of applause. And I just have one last announcement to make. This is a mini series on stretchable electronics and devices. And we're going to have part 2 of the mini series given by Takao Somiya of University of Tokyo. Again, it's going to be hosted by John Rogers. It's on Wednesday, August the 4th. And it will start even slightly earlier. So the seminar starts at 9 o'clock a.m. US Eastern Time, and the informal interview will start at 8:30. Until then, you all enjoy some of your rest of the day, and good night for Dae-Hyeong. Thank you again, and we'll see you next time.
Thank you very much.
Can I tell you guys a secret? It's Dae-Hyeong's birthday today.
Wow.
Wow.
Happy birthday.
Wow.
Happy birthday, Dae-Hyeong.
Happy birthday.
Oh,
I'm not going to tell
birthday.
them your age. I'm just telling you your birthday.
Thank you very much. Thank you very much. Yes. Thank you.
Okay, so I'll tell you another secret. I offered Dae-Hyeong a couple of different dates. He chose today.
To celebrate with us.
Thank you very much.
Great. Okay.
Thank you. Thank you all of you. Bye.
Thank you.
Bye.
Bye.
Bye.